Recording begins. It's April 29th. This is Biology 105. Thank you all for being here for another Biology 105 lecture. Okay, so far we've been talking in the last few lectures, we've been talking about DNA. We talked a lot about DNA replication and how that works, and the leading strand and the lagging strand and the replication fork and the enzymes that are present, and the Okazaki fragments and all of that stuff, okay? And now we're ready to get into transcription, okay, which is when the DNA is read and is used to direct the synthesis of RNA. Following this, we'll move into a discussion of translation, where the RNA is read and is used to direct the synthesis of a protein, all right? Are there any questions about anything in the course before? Oh, also, the lab practice final is now live, but it does cover material from this week's lab. It has a Hardy-Weinberg equilibrium problem, all right? So I suggest jumping into it after this week's lab, unless you're already totally good on Hardy-Weinberg equilibrium calculations. If you go to the lab materials, there's a worksheet that you can do. And if you do that worksheet and you're like, oh, I totally get all of this, then you could knock that thing out early. But that's your study guide for the lab final, right? It's basically the lab final, but in a simple, it's an online version, okay? I wouldn't say it's actually any simpler, all right? But you do it online, and it's worth 20 points, and you get 3 hours to do it. Any questions about that? Yes, please. No, the lab final is just on I think it's like a 6-page thing, but there's a lot of space for you to write out calculations and stuff. And that will be graded by your lab instructor. So if you've got additional, additional questions, you can always raise them to the lab instructor, but what am I saying? It's better to just ask me, because I'm going to give them the key and they're going to grade based on the key. It's pretty objective. There's not a lot of interpretation where there are questions where like, oh, I kind of got it right. It's like, nah, you kind of get it right or not. Cool? Good? Question?
So the lab final is going to be during our lab time?
That's awesome. Yeah, so the lab final is during your lab time next week. So your last lab of the semester, the entire lab period is set aside for doing the lab final. So you got 3 hours to do it, and it's really like I've said in the past, the majority of students finish it in under an hour, but some people take 3 hours, and that's not really a predictor of how well anybody does, so you can take your time on it. It's open lab notebook. Don't forget to have your lab notebook. Anything physical is allowed in the lab notebook. The only limitation to the lab notebook is 1, it can't be electronic, all right? And 2, your imagination, all right? Any other questions? Let's get into it then. We are talking transcription. Okay. So here's the central dogma in another form. All right. Remember, transcription, translation. What are the molecules? This also indicates some important differences between DNA, RNA, and a polypeptide sequence. Going for DNA is double-stranded, right? Bless you. And it's got A, T, C, and G. RNA is single-stranded, and it has A, U, C, and G. The other difference between DNA and RNA nucleotides is the sugar's different. So in DNA, it's deoxyribose is the 5-carbon sugar, and in RNA, it's just ribose, all right? So there's a slight difference in the structure of the sugar, and wherever there is a T in DNA, there is a U in RNA, all right? And there's a we in team. Bless you. And then in the protein, all right, the protein's not made up of nucleotides. The protein is made up of amino acids, if you remember from our biological macromolecules talk way in the beginning. Any questions about anything on this slide? We will talk about codons. Don't worry about that. Okay, cool. So when we get into translation, we'll talk more about this process, but let's just stick to transcription for now. I guess actually we're going to be, they overlap so much, we're going to end up talking about both. Okay. So you've done a green fluorescent protein transformation at this point in E. coli. It seems like most labs had success with that, which is really cool. So you've done genetic engineering now. That's pretty neat. You transformed some E. coli. You gave them antibiotic resistance and the ability to make green fluorescent protein. A lot of people went for the art project. I hope it went well and it turned out and you enjoyed and had fun. This is from a lab, 5 years ago or something. You can see that the image has been superimposed over another one, but somebody did that in this class. Okay? So, DNA. There are 4 possible nucleotides, A, T, C, or G. RNA. There are 4 possible nucleotides, A, U, C, and G. There are 20 different amino acids, all right? But RNA, well, DNA and then RNA encode these proteins. So it can't be a one-to-one, right? A can't just translate to a single amino acid because then you'd only be able to encode 4 different amino acids, right? So instead, you need this coding system to allow nucleotides, all right, to encode all 20 different amino acids. And so, I kind of misled you on the last slide. We're going to be talking about codons right away. All right, so here's the thing. In order to encode all these different amino acids, the ribosome, which is the organelle responsible for translation, it reads the nucleotides in units of 3. Think about a padlock, right? That padlock, the spinning one for your locker or something, right? It's got however many numbers on it. But it's not as simple as, my code is 49 and I spin it to 49 and then I'm done. You do 3 different numbers in sequence. And that means that by chance, you're basically not going to just by chance select the code because there's so many possible combinations. By breaking there's 4 possibilities for each nucleotide, and by grouping them into packets of 3, there are actually 64 different potential combinations for each 3 nucleotide grouping. All right? So each codon, which is a 3 nucleotide grouping, there are 64 potential codons. All right? Cool. And I'll show you how to do. The math there is 4 possibilities raised to the power of 3 in a block of 3. Okay? So there are 20 different amino acids, but there are 64 possible codons that you can make. So in this, it says, hey, how many nucleotides correspond to an amino acid? It's 3. All right? A codon is 3 nucleotides long, and each codon corresponds to a specific amino acid. All right? Now, there's 64 combinations and only 20 amino acids. So that means that either the codons aren't super specific or that there's redundancy built into the code. In other words, multiple codons can encode the same amino acid. So right here, you've got a codon table. And you're asked to be able to interpret a codon table on your final exam. A codon table looks a little intimidating at first, all right? But once you're oriented to it, it's not so bad. Yes, please.
Aren't there more amino acids that our bodies want to make, or is it 20 total across all?
It's 20 total, but some amino acids need to be supplemented. And so they're like vitamins, right? So you have amino acids that you need to consume that can be present, but remember that like this genetic code is, not remember, I haven't told you this. This genetic code is pretty universal. That's why you can take a gene from E. coli and put it into a plant or an animal, and it still makes the same functioning protein. Or take a gene from a jellyfish and put it into E. coli, and it still makes the same protein, because the ribosome works in the same way, all right? But yes, there are essential amino acids that we don't make in sufficient quantity that we have to consume in order to have enough building blocks to make protein. Yes, absolutely. But 20 is the number of amino acids. This is all that could possibly be made by a living organism. Some living organism must make those amino acids, right? We just eat it. Yeah. Sick. So, the codons are specific. There's not really so much flexibility within each codon, but there is redundancy. So if we look at the cell that I've highlighted with the laser pointer right here, you can see that the codon CUU, CUC, CUA, or CUG, they all encode leucine. They all encode the same amino acid. And if you look above that, also UUA and UUG, those all encode leucine. Now you might notice that in this codon table, there are U's instead of T's. That means that the codon table is used for the RNA, the messenger RNA, not the DNA sequence, okay? Let's orient ourselves to a codon table. Let's say that you've got a sequence of mRNA and you want to know what amino acids it's going to encode, all right? Well, not to get too far ahead of myself, but there's this first codon that sort of needs to be there and indicates the start of a protein. It's AUG, also called the start codon. So this is really great if you're doing genetic sequencing, because you can say, oh, there's a start codon here. That could mean that a gene is going to be downstream of this. All right? So anyways, you're looking at an mRNA strand, you find the start codon, you go, okay, that's the start of the coding portion here. Then you go to the next three bases. And let's say that the next three bases are the GAU. Well, then you would find on this side the first mRNA base, G, and then at the top, you would find the second mRNA base, A. You go down to the intersection of the two, and this is where all of the GAs are going to be. And then you can find the third RNA base on the right side, and then draw a line over there to GAU. Really, once you get the first two, there's only 4 options. So like, are you really going to go over here and be like, then I got to find out where the U is? You're just going to read GAUs right there. Okay? Yes, a question.
How does the start protein guide you anywhere on the table?
How does, so on the, yeah.
You just have G, couldn't you just go down to G first and then A and U? Why is the start protein in A big difference?
I'm sorry, I'm not sure I'm understanding the question.
Does the start protein indicate what column you need to start in?
The start codon only tells us if you're looking at a long strand of mRNA, the start codon just tells us like, this is where we're going to start reading. It sets what we call the reading frame. It's the first codon that's going to be read. I can actually clarify this if I maybe, do I have a strand, a sequence example? I don't. I can draw one on the board. It'll be helpful.
There was something about a reading frame, but it's cut off.
Yeah, we'll talk about the reading frame a lot more in a second, so we'll be fine with that. Let me show you, I think this will help. I'm going to go screen up, and then I have to clean up this mess up here. Projector blank, computer. All right. Cool. So some of the stuff's coming that I'm going to be talking about is going to have more elaboration in future slides, but I'm going to give you the full sort of deal right now. So let's say that we've got a DNA sequence, and I have to be strategic about how I plan this out. Okay, if this is the DNA template, all right, then we need to have an AUG, so this would be a T, an A, and a C, and then the rest of this doesn't matter. So A, G, G, C, T, A, C, G, U, T, T, A, C, G, A. That's fine. All right, so if this is the DNA strand, right, then RNA polymerase will attach here at some primer sequence, we'll talk about that later, and produce this mRNA strand. It's going to be complementary to the DNA strand. U, C, C, G, A, U, G, C, A, U, G, C, and U. So it's just like base pair complementarity with DNA the two DNA strands, right? Wherever there's an A, there would normally be a T, but because it's RNA, there's a U. Wherever there's a G, there's a C, vice versa. Cool? Wherever there's a T, there's an A, right? So this is our DNA strand at the top. This is our complementary mRNA strand. And now the ribosome will start reading this RNA right here, because AUG is the start codon. So this sets what we call our reading frame, because now, from here on, the rest of the RNA is going to be read in units of 3. I'm just going to put et cetera here. Okay? So if we unblank this, great. So AUG, start codon. And now we know where we're going to be reading the mRNA in order to figure out the polypeptide sequence. So in the protein, we've got methionine, and then CAA would be like, here's a C, there's A, CAA is GLN. Is that glycine? No, glycine's, yeah. GLY is glycine. And it's not glutamine, because that's GLU. It's an amino acid that starts with a G. Cut me some dang slack, I don't know all 20, all right? All you have to know is like, the abbreviation's fine. So GLN, all right? And the next one is UGC. So you start at U, you go over to G, you find the C, and that's cysteine, CYS, all right? So there's the protein, the amino acid sequence, right there. So I tried to kind of just lay it all out and hope that that answered your question. Did it? Yes. Sick. Okay. Cool. Good. Screen down. And then here, I'm going to actually just shift this text up a little bit. Shrink that font. Boom. So the reading frame is going to be super important, but it'll come up later, okay? The idea is, if this nucleotide is missing, well, then the codon is now UUG instead of UUU. But it's not just one amino acid that's going to be changed. If this nucleotide is missing, all right, all of these additional codons downstream are read in units of 3, so every codon gets shifted. We call that a frame shift mutation. The reading frame changes. It's going to come up later, but that explains the last bullet point here, okay? So instead of getting too far ahead, how do we feel now about interpreting a codon table? Good? Okay, awesome. Now, you'll be asked, like, to go from a DNA sequence to a polypeptide sequence. So don't forget the mRNA intermediate, all right? And you'll also need to know that reading of the mRNA begins on the 5' end of the mRNA molecule. That'll make additional sense, hopefully, as we talk about the process of transcription. But it's difficult to memorize this. I struggled with it when I was a student, of like, wait, so DNA and RNA synthesis has to go in the 5' to 3' direction, and then we read starting at the 5' end as well. All right, that can get confusing, but I'm going to explain it when we talk about prokaryotic gene expression. Question?
If there's a question like that on a test, will we be provided a codon?
No, you have to memorize this whole thing. Yep, and this is like 50% of your final grade is memorizing this codon table. That's a good use of your time. No way, you get a codon table, for sure. But it's a very fair question. And the codon table is this codon table. So it won't be like, oh, a new form of the codon table. Oh no, I haven't seen this before. It'll be this version, so you know, it won't be out of the blue. All right. There's a bacterial gene. The gene is 600 nucleotides long. How many amino acids long could it be? 200. It's a conversion factor dimensional analysis problem, or it's just a reasoning problem. There's 3 nucleotides per every amino acid, so the length should be 200. Now technically, this is annoying, but the stop codon, the point where it says, hey, stop making a protein, that doesn't encode an amino acid. So really this should be 199, but this is meant to be, just introducing you to the concept. Yes?
Does the start codon code for anything? So would it be at 198?
The start codon does. It encodes methionine. So basically every protein starts with a methionine. And of course, in genetics, they'll talk more about that and how that's handled. Yeah, but you'll see there are these codons over here that indicate like, hey, now stop making the protein. And they don't actually encode an amino acid. Yeah. So this is the P-gloplasmid. Now, I used to do a lot more in this class with plasmid maps and plasmid map interpretation, and last semester I kind of cut some out. So you haven't been exposed to these questions really yet. All right, so here's a P-gloplasmid map. The P-gloplasmid, this is the one that you used to transform E. coli. A plasmid map tells you like what's on the plasmid and then how long is everything. Bless you. Like how big is the plasmid? The P-gloplasmid is 5,371 base pairs long. The gene for green fluorescent protein, it runs from base 1342, or base pair 1342, to base pair 2062, okay? So how long is the coding sequence for green fluorescent protein? How many nucleotides long is it? Well, if it starts here and it runs to here, it's like if you went from mile marker 10 on a highway to mile marker 15, how many miles did you travel? 5. You take the end point, you subtract the start point. Nothing wild, right? So I'll just tell you that this is 720 if you do the subtraction there. So if the coding sequence for green fluorescent protein is 720 base pairs long, then how many amino acids long is green fluorescent protein?
240?
240, yep. And, of course, 239 actually, but 240. So if it's 720 nucleotides long, how long is the protein? 240 is the answer that you would give on any sort of exam like this, okay? But, all of our stuff's multiple choice, so it's nice and easy there. And then the last question, is if each amino acid weighs 110 Daltons, how large is the protein? This is my opportunity. I've got one last shot to try to get you to get super into conversion factors. I love conversion factors. They can change your life. They can change how you deal. They can make math a part of your life in a way that's not annoying, right? Because I had to take calculus, and I never understood it. I could not do calculus now, but this sort of math, it's great. So I'm just gonna go ahead and set up really quick. 720 bases per green fluorescent protein. All right, then I want to cancel out bases. I want to know how heavy this thing is, the protein is. So I want my final units to be Daltons. All right, bases per amino acid, and then Daltons per amino acid. So what I'm doing here, and I'll bring the camera over. Whoa! All right, what I'm doing here is I'm setting up a series of conversions to give myself an answer that is going to be in Daltons per green fluorescent protein. I know the bases per green fluorescent protein. I'm also going to know the amino acids per base. There's one amino acid per every 3 bases. And now I know that each amino acid from the slide weighs 110 Daltons per every 1 amino acid. Then my units cancel out. And once I solve this problem, I'm going to get an answer in the units of Daltons per green fluorescent protein. So listen, I was urged to get into these when I was in, like 100 level classes, and I didn't. I was like, I'm just going to do the math. Honestly, I'm just going to I'm not going to memorize this. I didn't learn the true value of conversion factors until I was in graduate school and I was doing pesticide application rates for the evil company Bayer. A lot of my PhD was paid for by testing their herbicides and pesticides, and I'm sorry. All right. Let me tell you a brief story of being out. I was out at 5 a.m. because the sun was too hot, wearing a Tyvek suit. You know those? It's like impermeable barrier because I'm spraying poison. You use these boom sprayers where you pick it up like this, and it has pressurized gas, and you have to walk at a given pace. You have to play a metronome in your ear so you're walking at the right pace to apply things at the right rate. And I would be doing that at 5 in the morning and just looking around and seeing grasshoppers like spasming on the ground. That was my life for like 3 years, every morning. So don't accuse me of not paying my dues or doing my part to make the world a worse place. All right? Anyways, those application rates are in things like grams per hectare, but you've got a liquid solution with a concentration that's in percent weight by volume. And so you have to adjust that to an application rate of like milliliters per square foot. It's ridiculous. But all of that is simple if you learn to love conversion factors. We're at the end of the semester, right? So that's it about this. You're not going to have any problems that you couldn't do without these. But someday you'll look back and say, I should have listened to John, just like they've all done. Good. Okay, this is how you would do the math for this question. Any questions about that? Dimensional analysis, conversion factors, they're great. Learn to love them someday or don't. Either way, I still get paid, so cool. Transcription is simple, thank goodness. DNA replication, you've got all those different enzymes that are occurring at the replication fork. It's so complicated, right? We've got a beautiful workhorse enzyme here in the transcription process, all right? Which is just one big mondo enzyme called RNA polymerase. It does everything. It separates the DNA strand, and it adds the new nucleotides. You don't need the RNA primer, all right, to be excised because it's RNA that this is making, all right? We'll talk about how this process initiates with promoters and all those things, but the actual molecular toolkit is really simple. It's like one piece, RNA polymerase. Question?
Because there are two strands of DNA, does it create two separate strands of RNA, or is it just one?
I think that I'm going to give you 6 John points for that question. That's a great question. It depends. So here's the thing. Each DNA strand can encode its own genes. There's plenty of evidence to show that you can have two DNA strands, and each one is encoding a completely different protein product, like opposite one another, right? The strand that is going to be transcribed at a given point is determined by where's the start codon, right? So this RNA polymerase is only going to go in one direction, and you can tell which way it's going to go because RNA, just like DNA, has to be built in the 5' to 3' direction. So each strand of DNA, get a little complicated here, but we're just going to keep going with it. You would be looking for the AUG from the 3' to the 5' end. It's a little complicated, right? But a start codon on this strand, it would have to start at 3' and be like AUG. And for this other strand, it would have to start from the 3' and be like AUG. Because you're going to still follow the rules of the anti-parallel nature and the fact that DNA has to be synthesized because of energetics in the 5' to 3' direction. That's still going to apply to RNA because it's also a polynucleotide. So that is definitely beyond what we need to really be worried about. But yeah. Did I answer your question? Can I provide more detail to your question, for your question?
It did, in a way.
Okay, I'll take it.
Yeah.
Yeah.
It answered it in a yes or no way, not much say that.
Okay, yeah. Can I provide any further detail? Can I do an elaboration?
Well, I'm not sure I have an idea.
If it comes back, you let me know, and we'll talk about it more. I even have a slide that talks about how cool it is that you can have different protein products encoded on opposite strands of DNA. So the RNA polymerase isn't doing both strands at once. It's doing one strand specifically based on where the start codon is. Awesome. Okie doke. Good stuff. Yeah, and remember, has to be made 5' to 3'. That's important. Cool? Yes? Okay. There are 3 stages to transcription. Initiation is when the process is initiated. In elongation, the RNA elongates. In termination, the process terminates. Not too bad, right? There's going to be exactly the same stages in translation. It initiates, then the thing that you're making gets longer, and then you stop making the thing that you're making. Initiation, elongation, termination. Now, what determines where the process initiates? That's different between the, oh my gosh, I did it again. Oh, and it's on recording. I do this every semester, where I get mixed up on start codons and start codons in transcription. So, listen, you know what I was just talking about? I was all excited about how the start codon's got to be here. Transcription doesn't use start codons, John. Somebody should have caught me. It's your fault for not, yeah. So I'll just push that off on somebody else. Transcription doesn't use start codons. That's translation that uses start codons. RNA polymerase doesn't use a start codon to start transcription, okay? Ribosomes find a start codon to start translation. So I'm very sorry. I'm sorry for those of you watching the recording. I'm sorry, to everybody out there, to the whole world, to the whole world. Yeah, totally my bad. Everything else still holds, how there can be, genes encoded on either one of these things. But what determines where RNA polymerase actually binds is not a start codon. So sorry. It's actually this promoter region. Okay? So we're talking about the process of transcription, and I have already spoken to you briefly about transcription factors. If you remember when we were talking about intracellular receptors and steroid hormone signaling and how, when the hormone binds to its receptor, it can act as a transcription factor. I told you how, that means that it's going to encourage the expression of a gene. So we've got these little promoter regions on the DNA, and these promoter regions are sites where RNA polymerase can bind. That doesn't necessarily mean that it's always going to bind there, all right? Usually it needs to have transcription factors present. Otherwise, you'd be expressing every gene in every cell that you could possibly express, because they all have promoter regions. There's a ton of regulation about whether the RNA polymerase is going to bind to the promoter or not. All right, so the 3 stages. In initiation, all right, RNA polymerase binds to the promoter, and the DNA gets unwound, and you start making RNA. You attach the first nucleotide. It's initiated now. In elongation, RNA polymerase is streaming down the DNA and building an RNA transcript in the 5' to 3' direction. In termination, the RNA polymerase reaches a sequence which, I'm going to go on a limb and just say I remember this correctly, is called the terminator sequence, which tells the RNA polymerase, okay, we're done now, fall off. And then it pops off, and now you've got a completed RNA transcript, which can be read by a ribosome, and that ribosome is going to start reading this RNA transcript wherever the start codon is. The start codon could be over here, This could be, this part of the mRNA, it could be that it isn't actually converted into a sequence of amino acids. The start codon doesn't have to be at the very end. Okay. Cool. So the promoter, all right? The promoter is just a region where RNA polymerase binds, okay? Promoters are characterized by this structure called a Tata box, which is really fun, all right? It's a sequence of adenines and thymines, right? Of A's and T's, okay? That is present in most promoters. RNA polymerase needs it to attach. There's other reasons to explain why it's there, but why does that matter to you? Well, somebody who's looking through a gene sequence, right? You do it with computers now. You're like, okay, we got a whole genome sequence of this organism, and now we want to find out where the genes are. Like, where are the parts of the genome that are actually going to be expressed and used to direct the synthesis of protein? So the computer scans for Tata boxes, and then it goes, this has a high probability of being a promoter region. And that's it. That's why it's important, okay? So promoters signal where you're going to start. They are what we would call upstream of the start point for transcription, because if this is the start point where the RNA is where you're going to begin attaching the complementary bases, all right, then you're moving from here in this direction. Oh, it's hard to do with this laser pointer. In this direction, right? So this is upstream, and this is downstream. Solid? Okay. Transcription factors. I've said it already, but if all you needed was a promoter region for RNA polymerase to bind, then RNA polymerase would be expressing or producing the mRNA for every single gene that every single cell could possibly make. and you wouldn't be able to have different cell types. It'd be energetically wasteful. It'd be silly. So instead, these transcription factors are factors that are required for transcription. And a transcription factor will sort of facilitate the binding of RNA polymerase to the promoter region. All right, so if the transcription factors are present, then RNA polymerase can bind. And yes, there are different RNA polymerases, because of course, it couldn't be as simple, as we've talked about, as just, there's just RNA polymerase. But don't worry about that for Bio 105. We'll worry about that later, okay? So if the transcription factors are present, RNA polymerase can bind and initiation can begin. So far, questions? Cool. All right, why did my font change? It's so annoying. This is called transcription initiation. This is called the transcription initiation complex. All of this right here. That's what that text says. Then you got elongation. All right, it just keeps moving along the DNA. It untwists it, and then it lets it snap back together. You don't need the topoisomerase because you're not separating too much of this at any given point, just a little portion. It does the work of the helicase. It extends. You don't need to know the rate of how quickly this happens. That's fine, all right? But it is interesting to note that a gene can be transcribed by multiple RNA polymerases at the same time. So you could have an RNA polymerase right here, and then an RNA polymerase right here, and then an RNA polymerase right here, all right? And then you're making more RNA transcript. So you're kind of amplifying your signal. The transcription factors attach, they allow RNA polymerase to attach, and then you get a bunch of RNA molecules, each one of which can be used to make protein, So you're making additional messages. You remember I'm the king in the castle and I'm writing the letter. It's like if I wanted to make a bunch of catapults, I would write the instructions of how to make catapults multiple times, make multiple RNA transcripts, and then send them to multiple catapult specializing villages. Okay? Very good. Termination, what do you reach? You reach the terminator sequence, okay? And then it falls off. The process stops. There's nothing too thrilling about this. You don't need to memorize these numbers. They're not important for this portion. Okay, they're not important for this level of course. Okay, we will talk about the polyadenylation signal sequence a little bit. Essentially, when transcription is ending, in the RNA, you end up, there's a bunch of T's, all right? And so the RNA ends up having a bunch of A's at the end of it. All right, it's fine. In prokaryotes, all of this is happening in the cytoplasm. There's no nucleus, right? So transcription can finish, or transcription can be in progress, right? You're making the RNA transcript, and as it's extending from RNA polymerase, you can already be attaching ribosomes. So transcription and translation can happen simultaneously. You can also attach multiple ribosomes to an mRNA at once in a structure called a polyribosome. This further amplifies a single gene. You've got the single DNA gene sequence right here, but you're getting multiple RNA molecules, all right? And each RNA molecule is being translated by multiple ribosomes, making multiple proteins. So you're taking a single set of instructions and you're making a ton of gene protein product. You're making a ton of protein product, all right? Now, this right here, this is the way to remember which direction ribosomes read mRNA in, all right? Because ribosomes are pretty similar between prokaryotes and eukaryotes. They're not identical, but they function in a similar way. Because the RNA must be made in the 5' to 3' direction, that means that this mRNA molecule, the 5' end is what's protruding. Everybody cool with that? So if transcription and translation can occur simultaneously, that means the ribosome must attach to the 5' end. Because the 3' end is not done. It hasn't broken off yet. All right? So that's the way to remember, like, where do I start reading for my codon table? It's like, oh, I have to start reading at the 5' end because a ribosome can bind before the mRNA is finished. And that means that they must be starting at the 5' end because the 3' end is not done yet. That's the way to, like, or you can just memorize transcription is, synthesis is 5' to 3'. Ribosomes bind to the 5' end. But memorizing things like that, it's pretty easy to get mixed up and confused when you got no way to fact check, yourself. Okay, cool, good. Moving forward.
Is that the same in prokaryotes and eukaryotes?
No.
Okay.
Well, it depends on which aspect. So the simultaneous transcription and translation is not the same in prokaryotes and eukaryotes, but the direction, which I think might have been what you were asking. Were you asking about, like, does the ribosome? Sorry, the answer to your question is actually yes. Yeah, the direction where that the ribosome reads is the same between prokaryotes and eukaryotes. So and that's how you can kind of remember it, of like, oh, it works in prokaryotes, you can do them both at the same time, and the ribosome's not that different between prokaryotes and eukaryotes, so the ribosome must read in the same direction in the eukaryotes as well. So, in eukaryotes, there's more steps, all right? There's a transcription step, and that makes this molecule that's called pre-mRNA. And then there's an RNA processing step where that pre-mRNA molecule gets modified, all right? Some pieces get chopped out of it, some pieces get shuffled around. You add this cap and this tail to the molecule, which are going to facilitate export from the nucleus and also make the RNA molecule slightly more stable, all right? There's all of these additional steps that are encompassed by the label of RNA processing. But that's what we'll talk about on Friday, okay? So apologies again for the start codon flub. I've been making that same mistake since my first semester here. And I've taught this class a dozen times now, and I still always fall into it.
It might be because you start talking about start codon before.
Yeah, no, I think I need to, like, really separate transcription and translation more clearly, and then I would stop getting mixed up. Yeah. But that would require extra work. I have to change something, and I don't change because I'm perfect. Yeah,
Yes.
yeah. Thank you for watching this recording.